Hello, good morning everyone. Today we are going to discuss about techniques used in parasitology. So first we discuss the concentration technique used in the parasitology. Significance of using concentration technique in parasitological diagnosis. So here if they ask any question, first you have to give small introduction about that. So now we have to give the what is concentration technique. So the concentration procedure separate the parasite from the specimen constituents and increase the chances of detecting parasitic organisms when these are in small number. That means the specimen received in parasitology normally fecal sample or the blood sample. In that the parasite concentration is very low. We cannot detect in direct method. That means direct method means wet mount. So when we are go for the concentration technique, that means we are concentrating the amount of parasite which are present in the specimen and concentrating, and we can collect the sample and the specimen, and we can observe, go through the identification. Here we are reducing the disturbance of the specimen constituent. For example, if the feces specimen feces containing fecal debris, that means sometimes it may interfere the identification of the parasitical stages, and sometimes if we consider the blood, when we consider blood sample, RBC, number of RBC is higher, so sometimes it will also interfere to identify the parasitical stages. So if we avoid these blood constituents or fecal constituents, that time we can identify easily the parasitical stages. So we can differentiate species wise, and directly we can go to the quick treatment if we identify the species. Then light infection can be detected by using this technique. That may be missing by using a wet mount. And here the concentration technique, we can perform feces and blood. Not only these two sample, other specimen also we can use this concentration technique. For example, urine specimen, if you identify Trichomonas vaginalis for your the laboratory, so if you receive the urine sample, so you can centrifuge it, and you can take a deposit and you can identify it. The centrifugation also the concentration technique. We will discuss coming slides. Right, when we consider the stool specimen, we have to use 3 consecutive samples. That means consecutive sample means we have to collect continuous 3-day sample. Not in single day 3 sample collection is not called as a consecutive sample, because most of the parasites are shaded their parasitical stages with different concentration. Sometimes when you consider first sample, that means first day specimen, sometimes you may get very low amount of sample, and sometimes second day you can get more numbers, or sometimes you can get third day only you can get the more the amount of the sample. So 3 consecutive means 3 continuous 3-day sample you have to collect. When we consider the stool sample, normally the concentration technique we are used most commonly Catocat's quantitative method, sedimentation method, and the floatation method. Now we will go one by one. Catocat's quantitative method is the advantages. This is the quantitative method. That means we can quantify the number of parasites present in the specimen by with the particular amount of the specimen. and can quantify the intestinal parasitical stages. Normally we use catalogs for the identification of egg. Especially we use catalogs quantitative method for Cystosoma mansonia. A sedimentation technique, this is very useful for the heavier egg that do not float in the salt solution. That means when we consider the eggs according to the weight of the eggs, some are deposited, some are floated. So in the case of sedimentation technique, here we are sediment the parasitical stages by using the specific gravity solution. So here the specific gravity solution is the compare with the parasitical stages lighter. That means low specific gravity compare with the parasitical stages. So the parasitical stages are deposited. So we take the deposit of the parasite and we can observe by wet mount or permanent stain or any other. Example of this method, the unfertilized egg of egg ascaris. Here these eggs are very weighted the parasitical stage. So by using flotation we cannot detect this stage. Other one larval stage of strongyloid and other one tinea egg. These are the example of the sedimentation method. The other one is the flotation method. This is useful for the light egg. In some cases, some parasitical stages are the weight is very low. So by using sedimentation, actually we cannot deposit these type of stages. So we go for the flotation technique. Example, protozoan cyst, thin ball nematodes. Other one, the second is most common specimen we receive in parasitology department, blood specimen. Here we use concentration technique, thin film, Knott concentration technique, membrane filtration, sedimentation method, the cytocentrifugation, gradient centrifugation. Thick film examination. Here identification of blood parasite like malaria, filaria and trypanosoma are the example. Here when we consider a thin film we are taking very minute amount of blood. That means very small drop of blood sample and we have to prepare thin film. But in the case of thick film we are adding more than 1 drops, the 2 or 3 drops. That means increase the chances of the parasitical stages. When we are taking more sample, sometimes there are more chances to detect the parasitical stages. Then Knott concentration techniques. This is used to detect the microfilaria only. Then it is also quantitative method because we are taking particular amount of blood sample and we have to do this procedure. So we can quantify. For example, if we take 1 ml of blood sample, out of 1 ml of sample, this much of parasite present in this parasite. So we can calculate the number, quantify. This is very accurate method. And membrane filtration, this is very cost effective because the membrane is the filtration, the instruments are very cost effective. So these also are used to identify the microfilaria. It's also quantitative method, can quantify number of microfilaria. Knott concentration technique, this is also one of the past paper question. So you have to know how to do the Knott concentration technique. These technique used to identify the microfilaria present in the blood sample. So here we need 2% of formalin, 10 ml. Then we have to add 1 ml of blood. Then we have to mix well and you have to centrifuge 500 gram per 2 minutes. Then after that you have to collect the sediment. You have to discard the supernatant and by using the sediment you have to prepare the smear, and directly you can observe the presence of parasitical stage, or you can stain with the Giemsa or Larval. Then you have to examine under microscope for the species identification and identify the microfilarial species. Membrane filtration technique, you should know the step, same like a notch concentration technique. Here you have to take the 10 ml of distilled water and 1 ml of blood sample. You have to mix it well, then you have to filter by using 25 millimeter membrane filter, and the pore size of the membrane filter is 5 micrometer. Right, it's actually, this is the membrane is like look like a transparent membrane. Right, then remove the filters, that means there is a filter, membrane filter, this is very transparent, and we have to take it out. Then we can directly go observe whether the parasite is present or not, or you can go for the staining, Giemsa or Larval, then examine under microscope for the species identification. Same like the filtration by using panel, right, and filter paper, right, but now we are using normally laboratories, the filter paper is the paper opaque, that means not transparent, but here the membrane filter is a very transparent, so you can easily observe after staining, you can easily observe under microscope also. The other important technique is culture techniques. Now you have to give the definition of the culture technique, right? The culture technique used normally when the specimen is negative for the wet mount and the concentration technique. Why we use the concentration technique? The wet mount is negative, that means the specimen is very low amount of parasitical stages. When we are observing under wet mount, saline in iodine, it shows the negative, but patient still has cyanide symptom. So we have to go for the concentration technique, because by concentrating concentration technique, we are concentrating the specimen contain low amount, we are concentrating the stages. Even if concentration technique is also negative, that means that much of low number the parasitical stages present in the specimen, so we can go for the culture. Even if a specimen contain a single parasitical stages, by using culture technique we can magnify, that means we are culturing, growing the parasite. So if it is, we see only one parasite is present, it will grow in a culture media, and we can identify by using further. Right, what is the use of these? We can use here artificial media or animal inoculation. In some cases we can use artificial media like microbiology. Some species they need animal inoculation, that's also culture technique. If the direct actually why we use culture technique, if the direct smear is negative, then we can go for the culture technique. And this is the other significant used in research work. Most of the time the culture techniques are used in research works. And antigen preparation, here by using the culture we are increase the number of the parasitical stages. So by using that stages we can prepare the antigen. For the ELISA kit preparation or ICT kit preparation, we can use them. Then larval culture, we can use the culture for larva, especially the identification of hookworm and strongyloids. Example, Harada-Mori technique. There are so many culture techniques available for identification of larval stages. Other thing, teaching purpose. Nowadays we are using the parasitical stages for the teaching purpose by using culture technique. can do for the blood and intestinal parasitology technique. Not only for the intestinal parasite, we can use for the blood sample also. Then studying of the pathogenicity. Here by using the culture technique we can study further the how it is infect the human, what happened in the infective site. We can, if we want to study, we need to culture it and we can go for further. And other thing, drug susceptibility test. This also very important, like a microbiology. We are culturing, then we are applying the antibiotic loaded disc and we can confirm whether the parasite is sensitive or resistant. Right, nowadays the plant materials are very familiar to identify the drug susceptibility test. Larval culture technique. Normally larval culture technique we are doing the larval culture for hookworm and the strongyloid. They are normally, when we are observing the specimen, in these two species larval stages present, but we have to differentiate these two larval stages because hookworm infections are very somewhat very it's somewhat very severe diseases because it causes severe anemia, because it's in the case of hookworm they are taking to lot of blood from the host cells, so it lead to severe iron deficiency anemia. So if we want to differentiate this and we have to treat the patient from the iron deficiency anemia. So we are culturing to identify the parasitical stages. Here we do Haradamori filtrate strip culture, petri dish culture, charcoal culture, Biermann method and agar plate culture. Then use of thin blood film. Normally for the blood parasites we use the thin blood film and the thick blood film. Thick blood film already we discussed. The thick blood film is we are using for the one of the concentration technique. We are concentrating the parasitical stages. But thin blood film, when we compare thick and thin blood film is more useful than the thick film. Thick film only we are talking about whether the parasite is present or not. Right? But in the case of thin blood film, here we are maintaining the red cell morphology also. So here by using morphological features we can differentiate the species. So now we discuss the uses or significant or important of the thin blood film. This is used to identify the blood parasites. Example, malaria, filaria, leishmania and trypanosoma. And here we can prepare and observe and compare the morphology of the parasite, not only morphology of the parasite but also the RBC morphology also. By using these two features we can confirm the species, can identify the species of malaria especially. Then stages identification. Stages also we can identify here. And disease diagnosis, by using the parasitical stages, red cell morphology, they can diagnose the diseases. For example, in the case of malaria, the Plasmodium falciparum is more severe compared with other three species, because this Plasmodium falciparum affect the brain or brain, and it causes the cerebral malaria, because it's lead to death also. So we have to identify the species wise and we can find out the disease condition also. Then we will move the concentration technique. We already discussed the concentration technique. The formal ether sedimentation. Here The advantages: sensitive to detecting the ova and the cysta increased by 8 to 10 times. When we consider the wet mount preparation, these sedimentation techniques more sensitive because we are concentrating the parasitical stages 8 to 10 times. And here the size and shape of the parasite is maintained because here we are using formalin. This formalin acts as a preservative or fixative, so the size and the morphology maintained. Other thing that the technique is inexpensive and easy to perform. Here there is no any special equipments and other things, so we can easily do in our laboratories. And other thing, fecal odor is removed. Normally in our feces sample there is a odor, so when we are using formalin, it reduces the odor of the feces. So another one, formalin, we already discussed it is a fixative or preservative, so it kills the parasite. So when it's killed, this is no risky to acquired laboratory infection. As a laboratory scientist, when we are handling live or living parasitical stages, it may be infected to the laboratory scientist. So when we are using these techniques, formalin kill the parasite, so no risk. Disadvantage of this method: when we are using formal ether, here the trophozoites are killed because trophozoites are the very delicate organism. So when we are using formalin, this is kill the trophozoite stages. Then saturated salt floatation method. Here advantages is useful for the light eggs and protozoan cyst, thin wall nematodes we can separate by using saturated salt flotation. Disadvantages: here we cannot concentrate the heavy egg, the weighted egg we cannot separate it. Example, unfertilized egg of ascaris, larval stage of strongyloids, tinea eggs. And other disadvantage: if we are keeping more than 20 minutes, protozoan cyst and thin wall nematodes, it gets collapse and become a deteriorate due to the high specific gravity of the solution. Then here we are using high specific compared with the parasitical stages. The specific gravity, we are using high specific gravity solution. This specific gravity solution, it is affect the trophozoite cyst and the nematode eggs, so it become damage the parasitical stages. So we have to observe within 20 minutes. Zinc sulfate flotation. Here we can use to detect protozoan cyst, light chain, helminthes. Helminthes means nematode eggs, nematode, trematode, chestodes, eggs are concentrated in here. The normally flotation means we are collecting the parasitical stages of is top of the solution. And here the disadvantage, we have to keep the specific gravity always 1.2. That is the disadvantage. Other advantages is we are using formalin. It's preserve the store and reduce the odors. Right. Okay. Thank you very much.